now, and I would like to ask first presenter for today, and that is Flaviana Marzano, to start the presentation, please.
Okay. Good afternoon to everybody. First, I want to thank the organizer for this opportunity to show you our work entitled Plant microRNA Can Control Cancer. Just a moment, sorry. Can control cancer gene expression through a sequence-specific targeting mechanism, the case of MALAT1 and NEAT1. Diet in human health is no longer simple nutrition, but it may play a pivotal role on cell status by modulating apoptosis, detoxification, and appropriate gene response to environment stresses. In fact, epidemiological studies suggest a role of fruit and vegetable in protection against several diseases. Nutrients can reserve or change epigenetic phenomena such as DNA methylation, histone modification, and microRNA regulation, thereby modifying the expression of critical genes associated with physiological and pathological processes such as embryonic development, aging, and cancerogenesis. MicroRNA are a class of evolutionarily conserved small non-coding RNA of 1924 nucleotides and lights that regulate gene expression mostly at a post-transcriptional level. They are known to be involved in control of different processes such as cell cycle, programming cell death, cell differentiation, tumor development, metastasis, and sensing of nutrient stress. In this slide, there are some papers that support the hypothesis that microRNA are resistant in murine and human district. In fact, in this first paper, the author showed that there is a Escherichia coli non-coding RNA can affect gene on can affect the gene expression in C. elegans. Moreover, in this paper, the author shows that there is some plant driver xenomere present in different human samples. In this paper, Zhang et al. Demonstrated that there is a specific plant macroRNA 168A that can regulate a murine gene. So all of this data demonstrated that macroRNA are resistant to enzymatic digest in gastrointestinal tract, are stable in animal serum, and are able to regulate gene expression in organisms that ingest them. On the basis of all these findings, we have carried out a pilot study to investigate the potential effect of edible plant macroRNA on the expression of human genes involved in cancer onset and progression, and elucidate the molecular mechanism and functional role. To achieve this goal, we used two complementary approaches, a bioinformatic analysis and a different biological experiments such as cell proliferation or transcriptomic profile using next generation sequencing and validation using RT-qPCR and luciferase assay. About bioinformatic analysis, we searched human macroRNA with a common seed with the plant macroRNA, so we used MIR-base, and we obtained 3,172 plant macroRNA that are function analogous to 1,606 human macroRNA. MIR-base was used as a reference database to extract human macroRNA and their target genes, experimentally validated by reporter cell, real-time, and Western blot. The list of predicted human target genes were analyzed for statistical enrichment in functional pathways related to cell proliferation, apoptosis, cell migration, and other cell functions that are known to be related to onset and progression of cancer. So finally, we selected 7 plant macroRNA that share the 5' region with seed region of 20 human macroRNA. In this slide, there are the 7 plant macroRNA which we find by in silico analysis to have a putative targeting activity on human oncogenes and tumor suppressor gene. We can find this plant macroRNA in Ziamice, Glycinemice, MAX, and Medicato truncatula. To verify if the selected plant macroRNA were able to modify the cell proliferation rate, we perform a CCKA proliferation assay. So for this reason, we transfected colon carcinoma cell line without p53 and colon carcinoma cell line with p53 with a plant macroRNA mix and with a control mix. So we perform a specific time course. The CCKA proliferation result demonstrated that there is a reduction of cell proliferation in each time. But in particular, we can see that after 72 hours after the transfection, we can observe a greater reduction in cell proliferation in both cell lines. On the basis of a good result obtained from CCKA proliferation assay, we want to investigate about the role of plant macroRNA on whole cell transcriptome in order to elucidate the direct or indirect action of the plant macroRNA on the cell. For this reason, we perform a next generation sequencing, and in this table there are the reads obtained from the sequencing. So, about the results, these are the graph about the results. If we observe the colon carcinoma cell line with the P53, we can observe that, the sample transfected with plant macroRNA mix compared to the control sample give us 336 deregulating protein coding and 14 deregulating long non-coding. When we observe colon carcinoma without P53 and we observe the sample transfected with the plant macroRNA compared to the control, we obtain 90 deregulating protein coding and 10 deregulating long non-coding. So, about the protein coding, the analyses are in progress, but a preliminary functional analysis shows an involvement of this pathway with a statistical p-value. When we analyzed the group of the deregulated long non-coding, we observed that there were two particular long non-coding, NEAT1 and the MALAT1. They appeared downregulated in both experiments, and they had a specific downregulation with a significant p-value. Long non-coding are a class of non-coding RNA longer more than 200 nucleotides. They have different action, transcriptional activation, repression, regulation of RNA splicing, sequestration of macroRNA. Some long non-coding RNA play important regulatory roles in pathological processes. In particular, MALAT1 and NEAT1 are closely related to various pathological processes ranging from diabetes, neurological disease, and complications to cancer, such as metastasis. First, after next generation sequencing, we validated the obtained results. In fact, we performed the real-time PCR, and we can see that when we transfected colon carcinoma cell line without P53 with plant macroRNA mix, we observe a reduction of MALAT1 and NEAT1 expression. Also, when we transfected colon carcinoma cell line without P53 with the plant macroRNA mix, we confirm a reduction of the expression of MALAT1 and NEAT1. So, to clarify the molecular mechanism, we searched the plant macroRNA binding site in the 3' region of the putative target long non-coding MALAT1 and NEAT1. In fact, we found that medicato truncatula macroRNA 5754 and glycinemax macroRNA 4995 seed perfectly matched with the 3' region of the MALAT1 and NEAT1. So, to verify if the selected plant macroRNA glycinemax 4995 and medicato truncatula 5754 were able to modify the cell proliferation, we perform CCK8 proliferation assay. For this reason, we transfected the colon carcinoma cell line with a specific plant macroRNA, and the results of the CCKA proliferation assay show that there is a degrees of cell proliferation when the cells are transfected with this plant macroRNA. So, to demonstrate that macroRNA 5754 and macroRNA 4995 are able to regulate the expression of MALAT1 and NEAT1, we perform the luciferase assay. For this reason, we cloned the putative wild-type and mutated binding site on the downstream of the luciferase gene in P3 in a premier vector, and then we transfected them into the cell. The luciferase assay data demonstrated that macroRNA 5754 are able to reduce the firefly activity, and also the macroRNA 4995 is able to suppress the firefly activity for NEAT1. So to confirm the specificity of this binding of plant macroRNA for the specific 3' region of MALAT1 and NEAT1, we mutagenized this region, and in this form we can see that macroRNA 5754 followed to suppress the firefly luciferase. Also macroRNA 4995 followed to suppress the luciferase activity. To confirm the data obtained on luciferase upon the whole MALAT1 and NEAT1 transcript, we performed the real-time PCR. In fact, we can see that when we transfected on colon carcinoma cell line macroRNA 5754, there is a degree of expression of MALAT1 and in both cell lines. And when we transfected in the colon carcinoma cell line macroRNA 4995, there is a degree of NEAT1 expression. To confirm that macroRNA 5754 and macroRNA 4995 are able to regulate the expression of MALAT1 and NEAT1 directly, we transfected the macroRNA 5754 and macroRNA 4995 in the colon carcinoma cell line. And then 24 hours later, after, we transfected the anti-macroRNA. This anti-macroRNA can inhibit the action of the specific plant macroRNA. So we can observe in these results that when we use anti-macroRNAs, there is an increase of expression of the long non-coding MALAT1 and NEAT1. Coherently, when we observe the CCKA proliferation assay results, we can see that when we use it, sorry? Yes?
Go on, there was, I probably just...
Okay. When we use the anti-macroRNA for 5754 and anti-macroRNA for 4995, there is an increase of cell proliferation rate. Yes, to understand if the if macroRNA 5754 and macroRNA 4995 cause the arrest of colon carcinoma cell line through MALAT1 and NEAT1, we sequenced the expression of this long non-coding RNA, and then we transfected the cell with the specific plant macroRNA. When we can observe in these results, we can see that, as expected, the silencing of MALAT1 and NEAT1 induced a reduction of cell proliferation. And when we transfected the specific plant macroRNA, we did not observe a further reduction of cell proliferation. So all this data demonstrated that macroRNA 5754 and macroRNA 4995 reduce the cell proliferation by acting mainly on NEAT1 and MALAT1. So with all of this data, we can conclude that macroRNA 5754 and macroRNA 4995 are able to regulate the expression of MALAT1 and NEAT1 long non-coding, indicating that the double exogenous plant macroRNA wouldn't interfere with human endogenous RNA, regulating their expression in physiopathological conditions. This result may offer the opportunities for biotechnological application in the agri-food research field for the production or selection of specific cultivars enriched of nutraceuticals for the treatment of many human diseases, such as cancer. Nanoparticles made of plant-derived lipids possess intrinsic therapeutic activities against specific disease, may be used also to efficiently deliver specific drug into animal cells. All of this data have been published on Frontiers in Genetics in these months. And finally, I want to thank my colleagues and thank you all for the attention.
Thank you very much. Excellent, Flaviana. Thank you very much. And now we have time for a few questions. Again, as yesterday, people can ask questions directly when they activate the microphone, or they can write questions also into chat box. I can read it, but now you can also read the chat box. So please. We can wait a little bit. If people are writing, it can take some time. Can you read it? There is one comment. How about interaction from Eric? Can you see it, Flaviana?
Yes, yes.
Okay.
How about interaction between a microbiome and human, and what you describe? Yes, everything is connected, because disease, I think that our results are the basis of, are the molecular basis for the interaction between the microbiome and the humans, because some paper in literature reports that we know that there are, if you eat fruit and vegetables that have specific plant microRNA that can regulate human gene expression, we know that probably this plant microRNA can regulate also the activity of bacteria into stomach or about the microbiome. Yes, yes, in fact, this is the first paper that I reported. Yes. Probably there is interaction between the metabolite products from microbiome and the plant microRNA that we ingest. Thank you.
Any other questions? Emiliana, Flaviana? If not, then I'd like to thank you very much, Flaviana, for your nice presentation.